Okay. I'm Dr.P.Kalaivani, working as Assistant Professor of Zoology in Mellala College for Women, Erode. Now I'm going to talk about the topic genetic engineering. Genetic engineering is nothing but the manipulation of genetic makeup of living cells by inserting desired genes through a DNA vector. In 1973, Stanley and Cohen and Herbert Boyer designed a methodology for transferring certain genes from one organism to another organism. DNA is a genetic material which contains all hereditary information needed to create an organism. DNA actually does not make organism. It only makes proteins. DNA is transcribed into mRNA and mRNA is translated into protein and the protein then forms organism. The specific region on the DNA molecule that direct the synthesis of proteins are called genes. By changing the nucleotide sequence in DNA, the protein formation is changed. Genetic engineering refers to the direct manipulation of DNA to alter an organism's characteristics in a particular way. It is used by scientists to enhance or modify the characteristics of an individual organism. Genetic engineering can be applied to any organism, from a virus to sheep. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. Now, let us see the tools which are required for genetic engineering. The genetic engineering or recombinant DNA formation requires the following tools. Restriction enzymes. The restriction enzyme is a protein produced by bacteria that cleaves the DNA at specific sites. This site is known as the restriction site. They are important tools for genetic engineering. They can be isolated from the bacteria and used in the laboratories. They recognize short and specific nucleotide sequences in the DNA known as the recognition sequences. When the restriction enzyme recognizes a DNA sequence, it hydrolyzes the bond between adjacent nucleotide and cuts through the DNA molecule. Now let us see the types of restriction enzymes. Type 1. These restriction enzymes cut the DNA far from the recognition sequences. However, they don't produce discrete restriction fragments, hence are of not much practical value. Next, type 2 restriction enzymes. These enzymes cut at specific positions closer to or within the restriction sites. They are exclusively used for DNA analysis and gene cloning in the laboratories. They generate two different types of cuts. Blunt ends are produced when they cut the DNA at the center of recognition sequence, and sticky ends produce an overhang. Type 3 restriction enzymes, they are multifunctional proteins. And then plasmids. Plasmids are used in genetic engineering to transfer foreign genetic material into different types of cells. The foreign DNA fragments is inserted into the plasmid and the recombinant DNA molecule is transformed into the recipient cell. The transformed cells are selected by the antibiotic resistance of the used plasmid. In transformation, a bacterium takes up a piece of DNA floating in its environment. In transduction, DNA is accidentally moved from one bacterium to another by a virus. The commonly used method for diagnosing genetically modified organisms are Western blot, ELISA, lateral flow strip technique, quality PCR, and DNA sensors. DNA ligase they are a critical DNA replication and repair enzymes. They have been widely used in molecular biology and biotechnology applications such as cloning and next generation DNA sequencing. Gel electrophoresis is a technique used to separate DNA fragments according to their size. DNA samples are loaded into wells at one end of the gel and an electric current is applied to pull them through the gel. DNA fragments are negatively charged, so they move towards the positive electrode. This slide shows an overview of how bacterial plasmids are used to clone genes. Let us now discuss about the process involved in genetic engineering. It includes preparation of desired genes, isolation of vector, construction of recombinant DNA, introduction of recombinant DNA into the host cell, selection and manipulation of recombinant host cells, and expression of cloned gene. Preparation of desired gene. The desired genes to be cloned are prepared in three ways. The desired genome is isolated from the total genomic DNA of organism. To isolate a gene, the genomic DNA is cut into many pieces by treating it with the restriction enzymes. The pieces of DNA are separated on the basis of their length using electrophoresis. DNA segment of desired length is separated from the electrophoretic gel and used for cloning. In the second method, the desired gene is separated from mRNA of the gene by reverse transcription. The resulting DNA is called as complementary DNA. In the third method of isolation, the desired gene is synthesized using a computerized machine called DNA synthesizer or gene machine. This machine can synthesize DNA of 1000 nucleotide length according to predetermined sequence. The number of copies of the desired gene is not enough for cloning the DNA is amplified using polymerase chain reaction. The desired DNA segment is known as passenger DNA or target DNA or donor DNA. Now let us discuss about isolation of DNA vector. The DNA used to transfer the desired DNA into a host cell or organism is known as vector. This vector, also known as cloning vector, carrier molecule, cloning vehicle. Here there are two types of cloning vectors, namely plasmids and DNA of viruses. Now let us discuss about construction of recombinant DNA. The successful vectors should have genetic markers for identification of recombinants and genes for self-replication in the host cell. A suitable cloning vector is chosen and isolated for gene cloning. The desired DNA is inserted into the vector DNA using a restriction enzyme and DNA ligase. The resulting vector DNA containing the desired DNA is called recombinant DNA or chimeric DNA or recombinant vector. Now we are going to discuss about the steps which are involved in the process of introduction of recombinant DNA into host cell. The recombinant DNA can be introduced into the host cell by direct transformation, pathological agent, liposome fusion, and direct introduction. Bacterial cells intake the recombinant DNAs in the medium by direct transformation. The pathogenic agents such as bacteriophages and agrobacterium pick up the recombinant DNA and introduce it into host bacteria or plant cells. The plant protoplast and animal cells pick up the recombinant DNA encapsulated in liposomes. The recombinant DNA can also be introduced directly into plant cells and animal cells by microinjection or electroporation and particle bombardment guns. The recombinant DNA integrates with the genome of the host cell and expresses a new character in that cell. Next is selection and multiplication of recombinant host cell. After culturing, the host cells are screened for the presence of recombinant DNA. The recombinant host cells are selected from non-recombinant cells by the following methods: antibiotic resistance, nutritional complementary defects, assay of biological activity, immunological reactions of gene products, colony hybridization. And by means of visible characters. The selected cell containing recombinant DNA is called recombinant or clone or transformant. The recombinant cells are mass cultured to test the purity of the product of cloned gene. Now let us discuss about the expression of cloned genes. The desired gene is expressed in the form of a protein. The protein is isolated and tested immunologically. The recombinant producing pure product can readily be used in the future. Genetic engineering breaks the species barrier. It allows DNA of two different species for combination. Applications of genetic engineering. Genetic engineering has applications in medicine, research, industry, and agriculture and can be used on a wide range of plants, animals, and microorganisms. In medicine, genetic engineering has been used to produce insulin, human growth hormones, follistatin, human albumin, monoclonal antibodies, anti-hemolytic factors, vaccines, and many other drugs. In research, organisms are genetically engineered to discover the functions of certain genes. Industrial applications include transforming microorganisms such as bacteria or yeast or insect mammalian cells with a gene coding for a useful protein. Mass quantities of the protein can be produced by growing the transformed organism in bioreactors using fermentation, then purifying the protein. Genetic engineering is also used in agriculture to create genetically modified crops or genetically modified organisms. This slide shows us the steps involved in the production of transgenic goat. Golden rice is one of the best examples for the transgenic plants. This slide shows the process involved in the production of insulin from Escherichia coli. Some oil-eating bacteria like Pseudomonas putida used in oil pollution control. Thank you.